Hello everyone, now let us discuss about anatomy of pancreas. Coming to the introduction of pancreas, as the name indicates, pan means all and creas means flesh. Pancreas means it is all flesh. Is a retroperitoneal gland. Pancreas is a retroperitoneal gland that is about 12 to 15 centimeters long and 2.5 centimeters thick. Coming to the location, it lies posterior to the greater curvature of the stomach, that is located in the curve of the duodenum. It lies posterior to the greater curvature of the stomach and is located in the curve of the duodenum. The major function of pancreas is it functions as both as an exocrine gland and an endocrine gland. So an example of both exo and endocrine gland is pancreas. Coming to the parts, there are mainly three parts: head, body and tail. Head is the expanded portion of the organ near to the curve of the duodenum. Body is the central body and tail is the tapering tail. Now coming to the functions of pancreas, as we have discussed earlier, pancreas has an exocrine and an endocrine function. Coming to the exocrine function, 99% of the exocrine cells of pancreas are arranged in clusters called acini, and they produce digestive enzymes. The exocrine function of pancreas is to produce the digestive enzyme. Coming to the endocrine function, 1 to 2 million tiny clusters of endocrine tissue are called as pancreatic islets or islets of Langerhans. Pancreatic islets or islets of Langerhans are the endocrine tissue of the pancreas. They are hormone secreting cells. Exocrine are the acini. They produce digestive enzymes, and endocrine part of the endocrine tissue of the pancreas is called as pancreatic islets or islets of Langerhans, and they produce hormones. Coming to the exocrine functional path, the pancreatic juices are secreted by exocrine cells into small ducts that ultimately unite to form two large ducts. The two large ducts are, the first one is pancreatic duct or the duct of Wilson, that is larger. The next is accessory duct or duct of Santorini. Pancreatic duct or duct of Wirsung is the larger duct, and the next is accessory duct or duct of Santori. These are the two ducts. The pancreatic duct joins the common bile duct from the liver and gallbladder and enters the duodenum, and as dilated common duct called the hepatopancreatic ampulla or ampulla of water. The ampulla opens into major duodenal papillae and the inferior to pyrolic splinter of the stomach. And the passage of pancreatic juice and bile through the hepatopancreatic ampulla into the small intestine is regulated by the sphincter of hepatopancreatic ampulla or the sphincter of OD. And the accessory duct, that is the duct of Santorini, leads from the pancreas and empties into the duodenum, superior to the hepatopancreatic ampulla. Here, this is the path for the passage of pancreatic juices or pancreatic enzymes. Now, coming to the composition and functions of pancreatic juice. Each day, pancreas produce 1200 to 1500 ml of pancreatic juice. It is a clear, colorless liquid consisting mostly of water, some salts, sodium bicarbonate, and several enzymes. The sodium bicarbonate gives the pancreatic juice a slightly alkaline pH, that is 7.1 to 8.2, and that buffers the acidic gastric juice in the chyme, stops the action of pepsin from the stomach, and creates proper pH for the action of digestive enzymes in the small intestine. This sodium bicarbonate, which is a composition of pancreatic juice, is responsible for maintaining a proper pH in the environment, gastric environment, for the action of digestive enzymes in the small intestine. Here you can see the various pancreatic polypeptide enzymes of pancreatic juice. The first enzyme is pancreatic amylase. The function is starch digesting enzyme. Amylase means starch digesting enzyme. The next is trypsin, chymotrypsin, carboxypeptidase and elastase. They digest proteins into peptides. Protein digesting enzymes are produced in inactive form and they do not digest pancreas itself. That is an important point. These are the active forms. The inactive form of trypsin is trypsinogen. So every enzyme has an inactive form and they are produced in inactive form only. And their function is to digest the proteins into peptides. The next enzyme is pancreatic lipase. It is a triglyceride, that is fats and oil digesting enzyme. The next is ribonuclease, RNA into nucleotides. It digests the RNA into nucleotides. The next is deoxyribonuclease. It converts DNA into nucleotides. Now coming to protein digesting enzymes, the trypsin is secreted in inactive form called as trypsinogen. The pancreatic acnar cells also secrete a protein called trypsin inhibitor that combines with trypsin formed accidentally in the pancreas or pancreatic juice and inactivates its activity. And when the trypsinogen reaches the lumen of the small intestine, it interacts with the enterokinase, which converts it into trypsin. Usually, in the pancreas, trypsin is secreted in an inactive form called as trypsinogen. And if in some way trypsin is formed, they have again feedback mechanism. The pancreatic acnar cells, they also secrete a protein called as trypsin inhibitor that combines with the trypsin formed accidentally in the pancreas or pancreatic juice and inactivates its activity. So this inactive trypsinogen that reaches the lumen of the small intestine, it interacts with enterokinase, which converts trypsinogen to trypsin. The enzyme which converts trypsinogen to trypsin is enterokinase. And this trypsin acts on the inactive precursors, chymotrypsinogen, procarboxypeptidase and proelastase. Here you can see a name pro is added before the enzyme. Procarboxypeptidase and proelastase to produce chymotrypsin, carboxypeptidase and elastase. Now coming to the endocrine function of pancreas. 1 to 2 million tiny clusters of endocrine tissue called as pancreatic islets or islets of Langerhans. They perform the endocrine function. Each pancreatic islet includes 4 types of hormone secreting cells. Each pancreatic islet includes 4 types of hormone secreting cells. They are alpha or A cells, beta or B cells, delta or D cells and F cells. The function of alpha or A cells is to secrete glucagon. Glucagon is secreted by alpha or A cells of pancreatic islet. Next, the function of beta cells or B cells is to secrete insulin. 70% of the beta cells, they secrete insulin. The next is delta or D cells. They secrete somatostatin. And finally, F cells, they secrete pancreatic polypeptide. Now, let us see the function of each hormone that is secreted by different cells. The first is somatostatin. It is inhibited by pancreatic polypeptide. Somatostatin activity is inhibited by pancreatic polypeptide. And the function of somatostatin is to inhibit the secretion of insulin and glucagon. It slows the absorption of nutrients from the GI tract. The next hormone is pancreatic polypeptide. And the stimulant effect is seen when meals containing protein, fasting, exercise or acute hypoglycemia. All these scenarios, they stimulate the release of pancreatic polypeptide. And somatostatin and elevated blood glucose levels. somatostatin or elevated blood glucose levels, they inhibit the release of pancreatic polypeptide. Coming to the function of pancreatic polypeptide, it inhibits somatostatin secretion, gallbladder contraction, secretion of pancreatic digestive enzymes. Coming to next hormone, that is glucagon. The synthesis of glucagon is stimulated by decreased blood glucose level, exercise and protein meal, whereas it is inhibited by somatostatin and insulin. And the major function of glucagon is to rise the blood glucose levels by accelerating glycogenolysis and gluconeogenesis. Glycogenolysis is nothing but conversion of glycogen into glucose in the liver. And gluconeogenesis is nothing but converting other nutrients into glucose and release of glucose in the blood. The function is to raise the blood glucose levels and release of glucose into blood. Coming to insulin. Insulin secretion is increased by glucose levels, ACH, arginine and leucine, glucagon, gastric, GIP means gastric inhibitory polypeptide, HGH means human growth hormone, and ACTH is nothing but adrenocorticotropic hormone. All these things, they stimulate the insulin release, and it is inhibited by somatostatin. The function of insulin is to lower the blood glucose by accelerating the transport of glucose into cells. That is glycogenesis, converting glucose into glycogen, glucose into glycogen, and then decreasing the gluconeogenesis. It accelerates glycogenesis and decreases glycogenolysis and gluconeogenesis, and finally increases the lipogenesis and stimulates protein synthesis. These are the functions of insulin. Thank you for watching. Please subscribe for further videos on medical coding and CPC training.